In today's video, we're going to talk about amino acids. So about 500 different amino acids exist in nature, but in humans we talk about only 20 different amino acids to grow and function properly. And 9 of these are classified as essential amino acids. So these are the amino acids the body cannot make, so you have to get them through your diet. And so this is how amino acids look like, right? We have an amino group, a hydrogen, carboxyl group, and a side chain, what we also call a functional group. And the functional group can be acidic, basic, polar, nonpolar, so on and so forth. And so this is what determines the specific amino acid, right? So here we have the backbone, right? And this is the functional group, which makes up the specific amino acid, right? Whenever you have a carbon bound directly to a carbonyl, we call this an alpha carbon. So this is an alpha carbon. We also say that this is a chiral carbon for all amino acids except glycine. So this means that carbon is bound to 4 distinct things. However, in glycine, the side chain here is a hydrogen. So if it's hydrogen, we have 2 hydrogens. So there's not 4 distinct things, only 3 distinct things. Another very important thing to understand here is the pKa value for the amino group and carboxylic acid. So for the carboxylic acid, the pKa value is 2, so that means it can behave like an acid and give a proton. And for the amino group, the pKa value is 9, so it can behave like a base and accept a proton. And so this we call amphoteric. Amphoteric meaning that it can behave like an acid and a base. So here we see Fisher projection of amino acids. We see an LS amino acid and a D amino acid configuration. And these are very important to know the difference between the two. And actually, the only difference really is that in the L amino acid, the amino group is placed to the left, and here the amino group is placed to the right. The same thing we see here, right? For some weird reason, evolution has favorized the L amino acid configuration. So this means that in the process of translation, we know that these are chemically the same composition. We see that, right? But they're just mirror images of each other. However, only the L amino acids are picked for making proteins, right? They are the only ones picked for making proteins. Sometimes, however, the D amino acid does get picked for proteins, and then when these are coupled with the tRNA, we have an enzyme called the DTD enzyme, which will bind to the D amino acid and make sure it doesn't function properly or even destroy it. And so in all circumstances, the D amino acid will not get picked for making proteins. And if it does, it will not work properly. So this is the one picked for making proteins. Now on to task number 1. What will happen in pH of 1 and pH of 12? So here we have this with iron, right? Where all the negatives equal all the positives, so neutral. So in pH of 1, we have an abundance of H pluses of protons in the surroundings, right? So this means that this will take up protons wherever it can. So here it will take up a proton, and the same thing goes for here. When the surroundings is very basic, or a pH of 12 or 13 or 14 or something like that, we have an abundance of hydroxys in the solution, right? So it will lose H pluses and deprotonate. So if you want to deprotonate this, you simply just have to deprotonate here. So H2N right here. Now they ask you in this task number 2, draw structural formulas for the following amino acids with charges that are quantitatively dominant at pH of 7.2. So we have alanine, histidine, tyrosine. We need to draw them at pH of 7.2. So let's try and do that. So here we have alanine, right? Now remember that the pKa value over here is 9 and the pKa value over here is 2. I want you to put this in correlation with the pH value of the surroundings, which we know is 7.2. and then you can know and determine if it will deprotonate or protonate further simply by comparing the pKa value with the pH of the surroundings. So over here, the pKa value of this is greater than the pH, so it will protonate, and over here it's the opposite, it will deprotonate. So in the case of deprotonation, it will lose hydrogen, so CO minus, right? Here, it will remain the same, and in fact, it will also remain the same here because this does not affect anything. And when we go on to histidine, here it's the same thing, right? We'll deprotonate. But what about this one right here? Well, we know that the pKa value for this is 6. And so because of of the pKa value is 6, well, the pH is greater than the pKa, so it will deprotonate. So NH plus here will deprotonate to N, or just nitrogen. Then what about here? Here, we'll see deprotonation right here. And then the pKa value for this thing right here, which we call phenol, is 10. And so that makes it, so it will protonate. So the only thing happening here is a plus right here. And this is exactly the answer, guys, as you see right here. Now let's quickly talk about peptide bonding. So peptide bonding is due to condensation reaction and dehydration synthesis. So what happens is we have removal of water when monomers join to form a polymer. So here we have two monomers of alanine, right? They will remove water right here and form a polymer, right? So the OH and H gets removed as water. We get this peptide bonding right here. In reality, this is a nucleophilic attack on carbon right here. From nitrogen will make a nucleophilic attack on carbon. It's a bit of a complex process, but I will not go into depth about it right now because I don't have time for it. When we talk about peptide bonding, it's very important you understand that there's resonance, right? Sometimes, they usually write it up like this, but in reality, peptide bonding is not a single covalent bond only. It is a resonance. This double bond switches back and forth between the carbon oxygen and carbon nitrogen right here. It's very important to understand this because it makes it a 1.5 bond. So this is a bond that is stronger than the single bond, but not quite as strong as the double bond. Also, because we know that in a single covalent bond, we have free rotation, but because this is not a single covalent bond, so in reality, you don't see a free rotation. So it will become planar. We don't see free rotation. And this is very, 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 very important for the side chains of polypeptides, right? So here we have the side chains, right? As you see, it's a trans configuration. If the side chains were to only here or only to be here, we'd say that this is cis configuration, but this is trans configuration. And the trans configuration here is fixed in these positions due to what? Due to the resonance, right? And this is also very important because if it were to be in cis configuration, we'd see large side chains and they would likely crash into each other, right? Which would completely destroy the tertiary structure. So this is very important in keeping the structure of the protein. Now they ask you to draw the modes between which resonance occurs in a peptide bond, and I've done that here. So here you see the resonance, right? Here we have the double bonding here, and then we have the double bonding here. So this is an example of that. Remember, when you draw up peptide bonding or polypeptides or anything like that, make sure